Have you all here today. My name is Don Andrus. I'm the CEO of Novita Nutrition, and we're very pleased to have the opportunity to sponsor this webinar with Dr. Hanigan. And we very much appreciate your participation today. As many of you are aware, you're aware of Dr. Hanigan, his impressive research over many years. But for those that are not, I thought it might be good to provide a little background. Dr. Hanigan began his career as a dairy farmer in Western Iowa, followed by a BS in dairy science from Iowa State University, an MS in animal science, a Ph.D. in nutrition, and post-doctoral work and in biochemistry and biophysics, all from the UC Davis. He joined the research group at Purina Mills in 1993 and moved to the Department of Dairy Science at Virginia Tech in 2005. During his career, he was focused on improving our understanding of energy and protein metabolism, and his experimental work has been interfaced with mathematical model development. And his current research is directed at assessing individual animal responses to amino acids and protein and improving our understanding of the regulation of volatile fatty acid production in the rumen and energy metabolism in the animal. The goals of this work are to improve prediction accuracy of the current animal models for use in precision nutrition, and success in that endeavor will improve animal efficiency and reduce environmental impact of food animal production. He has mentored 28 graduate and postdoctoral students. He's the co-author of more than 170 peer-reviewed research publications, and was a member of the NASEM 2021 Dairy Nutrient Requirements Rewrite Committee. I recently had the opportunity to spend a couple of days with them and other leading dairy nutritionists at an offsite summit where we're focusing on the use of optimizers as a part of dairy diet formulations. And at the meeting, I shared with him a comment that he'd made in a previous presentation that really stuck with me. His statement was that dairy cattle are only about 25% efficient at converting feed nitrogen to milk protein. And so just a minor improvement would be very beneficial to the dairy industry. But before turning it over to Dr. Hanigan, please note the webinar is being recorded so others can listen to it at a later date. And Dr. Hanigan will be taking questions after the presentation. But because we have so many joining today, we have muted all mics. But so you can submit questions within the Q&A chat window, and then he'll respond to them following the presentation. So with that, Dr. Hanigan.
Thank you. Appreciate that, introduction. And you could probably also sum that up and just say, well, he's done quite a few things, and how come he keeps changing all the time? But let me try to share my screen here so we can, I'm old, so I can't remember what I need to talk about. I have to look at what I'm going to talk about. So there we go. All right, so you should hopefully see my presentation window now.
It is up, yes.
All right. And, he said all those nice things about me, but honestly, not much happens in a research group and in an academic setting if you don't have grad students and postdocs to work on things, because I just sort of become a money manager and, and file reports and things like that. So I want to recognize, a string of students who've worked on that. Jacqueline Prestigard, Peter Yoder, some of you probably know him because he's back in industry now. Gianna Daly, who's going to be joining the University of Missouri here shortly. She has been at Purina Mills. Vinicius Souza is back in Brazil, and Leticia Campos is in a postdoc position at the Colorado State University, and she'll be taking up a faculty position there. Also, none of this happens without money, and it seems like it's become popular to, I guess, diss government for, wasting all this money. Well, all of this research, that I will show you today really is based on 4 different grants that we've gotten over time, which, sum up to probably almost $2 million, all from, we've gotten industry funding as well and other funds, but these core grants, provide the core money that we need to run our program and keep it running. And so, if you don't feel like this is worth anything, then yeah, I guess the government wasted money on it. But if it helps you, then it wasn't a waste of money, and it's how we actually move things forward. So I want to talk, broadly about amino acid nutrition today. And those of you that have seen me talk before have probably seen one or more of these quotes. And I think I can sum up the quotes. These are from Mark Twain, an author, Will Rogers, a humorist, and Bertrand Russell is a philosopher, basically. And essentially what it boils down to is probably what Will Rogers is trying to say is that, there's lots of things we learn in life, and we forget a lot of those, but we also retain some of them. And the ones that we retain, not all of those are true. And that's the ones that really give us trouble, right? According to Mark Twain, it's not what we know, it's what we know that isn't really true that gives us trouble. So we have to once in a while look back, according to Bertrand, and hang a question mark on things and see whether or not when we reassess them or reevaluate them, are they really true? And this is one that, I'm sure probably everyone has seen because I've visited about 60 different countries in the world, and I don't think there's a single one that when I've talked to students on nutrition that they haven't related to this barrel and stave. Okay, so I wish that I could actually come up with some analogy that was this widely known and disseminated in the world. That would probably be the biggest contribution I could make if it were true. Okay, and the problem with this one is that it's a nice way to summarize and think about it, but it's actually not true, okay, in general as applied to animals. And I think it's probably not totally true as applied to plants either. It originates from the work of Sprengel, who was a German scientist in 1828, and he was a plant person. So he simply observed that when the soil was limiting in a particular nutrient, let's say it's limiting in nitrogen for the case of our example here, that plant growth would be less than optimal. And if he provided additional nitrogen to the soil, then the growth of the plant would respond proportional to the amount of additional nitrogen that he provided. That's true, okay? I mean, that's still true today. It was true at that time. It's also true for animals that when we have a nutrient that's limiting, if we provide more of that nutrient, the animal will generally respond. Where we started to go awry then was about 30 years later when a subsequent student in Germany decided that, well, it's actually more constrained than that, in that if a nutrient's limiting, then growth can't respond to another nutrient that we provide. And Whitson and Harlow then sort of took that concept and they came up with the analogy of this barrel and stave. And of course, it's sort of set up for plants here because it's talking about soil fertility. But in this case, the lowest stave, which is nitrogen in this example, is what's limiting the yield of those plants. And according to von Liebig, then there's no reason to add anything else, right? Adding potash or adding lime or adding, anything else won't make those plants produce more because they have a sole limitation of nitrogen. And this eventually became known as the law of the minimum, even though it's really just a theory, okay? It's not really a law. We do have laws of conservation of mass, which state that the plant can't put out more nitrogen than there is nitrogen that it can get from its environment. But that's not how we're operating here. We have more nitrogen available. The plant is just not 100% efficient at being able to take it up and use it. And so Mitchell and Block then sort of took that, that building that Whitson and Harlow constructed with the barrel and the stave, and they put a big patio on top of it when we started learning about all these amino acids in the 1940s. And they use that to define the order of limitation. So not only do we know which one's limiting, but we could tell from the calculations which one's second limiting, which one's third limiting. And so that would help us then construct diets. And it's useful, and it did help us construct diets. But the one underlying assumption to this that causes us trouble, okay, going back to Mark Twain, is that it assumes that there's constant efficiency, that the nitrogen efficiency of use for these plants, I have no idea what a plant, uses for efficiency, but let's say it's 40%. That is 40% under all conditions, that it never changes. It doesn't matter how much water's there, it doesn't matter how much potash, it doesn't matter how much oxygen, etc. And that is the required assumption for this whole idea of the law of the minimum, as von Liebig called it, to work. So what I want to show you is that looking at, some summary work that Helen LaPierre did in support of the NASM, when she looked at from the literature, a whole lot of, I think we ended up with, what was it, 230 studies with, about 600 treatment means. And she looked at those in terms of the MP supply that was provided versus the efficiency of conversion of MP to milk protein. You can see that essentially as we add more MP to the diet, the efficiency of conversion goes down. But, we've got some outliers at the top. We're sort of looking probably at about a 0.8 as maximum efficiency. In other words, 80% conversion efficiency. But of course, we range down to, less than 50. And so as we add more MP, then almost all of our diets are operating down in that 50 to 60 range. And she also was, a lot of Europe will ratio their MP to DE or to ME to try to provide a protein to energy ratio. And of course, the swine and poultry people do the same thing. And it tightens up the data a little bit, but it doesn't change the relationship at all. There's a whole range of this ratio that can be used. And the higher we go in the ratio, the lower the efficiency gets, okay? So that says that there isn't constant efficiency. And here's some more recent data from Letitia, who I indicated was at Colorado State. So she's got some studies to publish here yet, and these are not done. And so these are preliminary data, but essentially we looked at feeding 5 different levels. I mean, she's got it marked here as predicted MP intakes, but that's not every animal ate exactly the same amount. So the diet concentration of MP and CP is below that corresponds. So as we go from about 8.45% MP up to 9.74, we get an increase in milk protein output. But its efficiency is falling off, right? We get a straighter, a greater increase, a greater marginal return on that first increment of MP than we do on the second, than we do on the third. And of course, then the last increment gets no response. And so this is sort of what we expected, right? It's what we could see in the NASM data as well. But when you look then at individual cows on those treatments, we have, about a third of the cows that had linear responses. In other words, they never did get to their maximum production. We have more than a third, probably almost half, that did exhibit almost exactly the same plateau response. And then there was about a quarter of them that said, I don't really care how much protein you put in my diet. I'm going to keep making the same all the time. And so there's, of course, diversity in cows based on their genetic background. But the general trend on cows is we've got declining responses or marginal returns, which means we have falling efficiency as we go up higher and higher in MP. And you can see that on each of the individual amino acids. This is from the NASM publication. Lots of variation. Some of that's probably due to varying energy. But in general, you see, and particularly if we look at the methionine and lysine, which have larger ranges because of all the rumor-protected amino acid feeding studies and infusion studies, you get basically a hump, right, or a plateau in the middle and declining efficiencies at both ends. We decline at the upper end for the same reason I just stated for MP, in that the ability of the animal to convert MP to milk protein falls off as we provide more and more supply of these amino acids, which is really what the MP is, right? We don't really have an MP requirement. The cow has an amino acid requirement or a number of amino acid requirements, and we simply cover those by providing enough protein, enough metabolizable protein. But we can also muck it up on the other end, right? If we get one of the amino acids that's more deficient than the others, then efficiency falls off again, okay? So for example, maybe the back end of this lysine is because methionine was deficient, right? So, and of course our supply is less as well. And so the efficiency drops off as we start moving away from the center. So this concept was used really to set the target efficiencies for the NASM, and we'll come back to that later. But you can see that the same pattern exists for all 10 of the essential amino acids. So not constant efficiency. Now, why is it not constant efficiency? Well, there's probably about 5,000 research publications and 30 years worth of work, and probably several $1 billion spent on eliciting all the reasons why protein synthesis is not linear, okay, in terms of response to the supply of amino acids. And I've summarized, some of this, the core parts, that, from a variety of different publications, including some of our own, into this schematic. And this is actually a pretty big simplification. There's additional complexity here. Why is there so much complexity in terms of regulating protein synthesis? Well, it's a really expensive process, okay, for the cells. And it's also like an assembly line where, the proteins may be 2, 3, 4, 500 amino acid long chains. And so there's no sense starting making a protein that's going to require 500 amino acids if we can look down the sequence and see, oh, I don't have enough lysine. And so when it gets to its first lysine, it has to abort because it can't find a lysine. So it wastes all that energy, right? So it has lots of regulation because of that. And the regulation starts with amino acids, individual ones affecting a variety of these signaling proteins, but they almost all converge on the mammalian target of rapamycin, okay, which is a drug. And it got named for that because we didn't know what the protein was, but we knew this drug had this dramatic inhibitory effect on protein. So if there's adequate amino acids, it stimulates mTOR, and mTOR then, through taking off the brakes on 4-ABP, stimulates translation. If there's adequate energy in the cell, signaled by the AMP kinase levels and ATP levels, then that also impacts on mTOR. Okay, and it's all negative, where if there's not enough energy, it shuts it down, and if there's adequate energy, it lets it run. And also there's insulin and IGF-1 signals that are arriving from the rest of the body, which is stating, hey, we got enough energy in the body in general. We're putting away energy in body fat, so there's no reason you can't make as much protein as you would like to. And so that also impinges on mTOR. So these things all come together on mTOR and say go, no go, or go fast, go slow. Okay, it's not really a switch, it's a gradient. Okay. And so again, it's like an assembly line, and it's just looking at all the parts and the power supply, which is the energy, and saying, do I have enough of everything? If so, go. Go as fast as you can with given the amino acid supply you have. And so that also then is regulated. The uptake of amino acids has feedback inhibition. So if we have an excess of supply of amino acids arriving at the tissue and getting into the tissue, as they build up because they're not being used, they slow down transport. So that's what causes that curvature. If we keep chucking more and more protein into the system, the mammary gland starts resisting it more and more and more, okay? And the efficiency or the marginal returns on each investment of additional MP or amino acid become less and less. So this is it's more important, the regulation is, than the actual grams per day, okay? So we have to have all the amino acids, we have to have enough energy, and we have to have signals from the rest of the body that it's not stressed. I mean, there's other signals like stress hormones. If you're stressing the cow because of heat or because of poor environment or uncomfortable stalls, those signals propagate through this site as well, and they will slow down milk protein synthesis. So this variable use and variable transport creates that a variable efficiency of use. So it's not linear, it's not fixed, okay? That barrel doesn't actually capture the biology. Now, all that work was primarily done in cells, and I was a dairy farmer too, and so when somebody showed up at my door and said, "Oh, I got all this neat cell culture work," well, that's not a cow, okay? Well, it's also really hard to do this work in cows, and so while we were trying to get this done in cows, we actually collaborated with a group of colleagues, and this student, the UMA, did this experiment with lactating mice. And we used litter weight gain as a proxy. And so she first did a whole response curve and found that milk, or the litter weight gain, was maximized at about 21% crude protein in the dam's diet. And it was significantly reduced with 15%. Now she did from like 6 to 25, so she had the full range. We picked these two as a positive and a negative control, and repeated with this amino acid study. So you can see we had a protein deficiency. We added to that low protein diet isoleucine, and we got a significant response in pup growth. If we, instead of adding isoleucine, we added leucine, we got a significant response. If we added methionine instead of those branch chains, we got a response. The only one we didn't get a response to was threonine. So a single diet was limiting in 3 different amino acids, and we could provide any of those 3 and get a response. So von Liebig's, law of the minimum doesn't apply because there's variable efficiency and there's regulation of that efficiency in these animals. So not a valid, way to look at it. Okay, we can add more than 1 amino acid and expect to get a response. Can we just add any amino acid? No, there are some that are apparently provided under this situation in adequate supply to fully cover any maximal needs, probably. So we finally got it, sort of recreated with Peter Yoder's experiment that was published in 2020. So we had cows that were milking, over 100 pounds a day in metabolism stalls, making about 1,475 grams of milk protein a day on a 15% diet, and the gross nitrogen efficiency was 38%. Okay, we then, to those control diets, we infused methionine, lysine, and histidine, and got a significant response in milk protein output. And then on the next treatment, we infused, instead of those 3, we infused isoleucine and leucine. Again, we got a significant response. And in this case, we tried the 2 groups together, which we didn't do in the mice. And you can see that the response was even greater. In fact, when you take the 2 marginal responses, they almost perfectly add up to the marginal response to adding all 5. So independent responses and generally additive, and that's statistically supported by the lack of an observation of an interaction with them. Each one had an independent effect and they add together. There's no interaction to make it worse or better. So that really informed us in terms of the NASM work, right? We already knew the cell culture stuff. We knew all the molecular biology stuff, but the animal work, sort of sealed the deal and said, okay, the representation can't be this first limiting theory. It has to be an additive equation with at least histidine, and this was worked out through all of our modeling work: histidine, isoleucine, leucine, lysine, methionine. Threonine's there. It's not as well defined, but certainly it seems to be important. A huge impact of energy in the diet. And the energy that comes from NDF is not quite as valuable as the energy that comes from fat and starch, okay, and protein as well. And they do curve over at the top. So we got a negative sort of term, a quadratic term, which means that you can't just keep chucking in these things forever and expect a linear response. It does plateau. But where we're operating with our cows of such high genetic merit and our diets that are, more marginal, we can see some curvature, but it's not great. But we are certainly operating in that curved range, okay? If we drop down lower, we tend to see then greater and greater marginal responses. And this equation, this approach, halved our error of prediction and removed all of the, particularly the slope bias we had before. The old equations are terrible, okay? They overpredict responses in both directions by about 1/3. So they were not good. They went through the middle of the data, and that's about as good as you can say, okay? If you had 80-pound cows, it was going to hit the target that you needed for an 80-pound cow. If you wanted to try to get to 90-pound cows, you would have to go for almost 100 pounds of milk just to get them to get to 90, because that model was off by that much. All right, so how about, then coming back to these ideas of is there other amino acids? And so we had an opportunity with Letitia's experiment after we had done the protein titrations to come back with those 50 cows and then look at 2 groups of amino acids. So we did 2 more blocks, 25 cows on each amino acid, looking at dose responses to the addition of 3 additional amounts of each of these rumen-protected amino acids. And we had assessed all of these previously using our isotope approach, so we knew pretty closely what the metabolized value is for each of these. So the first one's histidine. And I want to point out again, preliminary data, okay? She finished her dissertation, but we have not gone through this data. And in looking at this again this morning, I'm pretty sure on the histidine, the first 2 treatments are reversed, okay? I went back and looked at her dissertation and her tables. This is how it is in her tables. But these cows were randomly assigned to treatment, and you can see that all the controls have roughly the same level of production, except the histidine cows. And this second bar here is almost right where it should be. So I think she's got these two mislabeled in her data, and we'll get that sorted out before we publish. But nonetheless, if we ignore that first bar, we got a response, almost a linear response to histidine. We got a linear response to isoleucine. Something happened in this third treatment with lysine again. Maybe we'll find something in the data, I don't know, but we got a response to lysine, but it wasn't really linear. And we got a linear response to the first two increments of threonine, and then we sort of, failed to get a further response. So threonine's new, right? We don't really have any data in animals on that. And we couldn't figure out whether it was isoleucine or leucine. We got responses to the combination, but previous efforts to look at both and delineate between had not really paid off. So this is a nice way, I think, to look at these additional amino acids, provided we can get people to protect them for us, and then we can assess what their protected value is. But again, it's all one diet. There's no different base diet here for zero. We added histidine, or we added isoleucine, or we added lysine, or we added threonine, and we got a response to every one. Okay, so independent responses. I can't tell you they're additive because we didn't do all of them together, but I can tell you they are independent responses, consistent with what the NASM representation has, and inconsistent with von Liebig's law of the minimum. So how do we, if we move then to this, what the NASM model provides for you in terms of diets, and try to do a little example here to show, what you can actually do with this. All right. So I took Bill's, high cow diet as is. It's corn silage, legume silage, some grass legume silage mix, some corn grains, soybean meal, protected soy, some cottonseed, distillers, some fat. I don't think it's really lard, but he chose that. But I wouldn't recommend feeding lard. Okay, that's too unsaturated. It's not going to be a good source for you. But anyway, and then some minerals, and then for the last one, I'm going to add some amino acids. All right, so I basically took a 17% protein diet, and I wanted to get it down to about 15. So I took out legume and grass, mostly legume silage, and added in corn silage. I left the soybean meal. I took the soybean meal down and added some protected soy. I took a little bit of cottonseed out. I added a little bit of distillers. I doubled the fat, basically. Again, don't do this with lard, but you can do this with a really, a good tallow source or some protected fat. And then for the third one, I left the crude protein the same, and I added in amino acids. I added in some room-protected histidine, lysine, and methionine. So what did I do for nutrients? I took the crude protein down like I just showed you. I took the MP down a little bit. Okay, that wasn't really my intent, but of course, as you move these things around, and drop that much protein out of the diet, you're going to lose a little bit of MP possibly. I added ME to the diet, or NEL, however you want to look at it. They're almost 100% correlated, so it doesn't really matter which one you look at. And everything else, well, RDP went down, but most of the other things are very similar, okay? I left the NDF the same, the ADF's about the same. I did add starch, okay, which is where some of the energy is coming from. So what's the model say? Oh, and so I'm sorry, for the last one then, no, let me back up. I got all those covered. Yeah, for the last one, then I just added histidine, I added some amino acids, okay? Yeah, I skipped a slide. So when we look at that first diet, the high protein diet, it's pretty close to the target efficiencies. And these are target, efficiency of use of total MP. Okay, so MP going into the animal, it goes to maintenance, it goes to reproduction, it goes to growth, and it goes to milk. This is for all of those, okay, that 75%. Most of it is due to milk. But we're just a little bit under or over the target for histidine and a little bit over for methionine, okay? When I take it down, when I take MP and crude protein down, now I've got a bigger problem with histidine, and I'm farther off the mark. Well, I'm now off the mark with lysine, and I'm farther off with methionine. I'm a little bit off. I mean, I would encourage you, don't worry about one or two, okay? They aren't that persnickety in terms of their supply. And so then if you look at the last table where I added in the methionine, histidine. What did I add in? I forgot now. Added in, yeah, methionine, lysine, and histidine. So I fixed my problems, right? Now I'm at or around the target for the histidine. My lysine is right at target. My methionine right at target. I'm a little bit shy still on threonine, tryptophan, and valine, but of course, those are not generally available in the marketplace. And whether or not 4% units is enough to worry about, I don't know. But certainly, if I was going to add something else and I had it available, I might consider doing that. Of course, it depends on the cost. So now, what did I end up with in terms of predictions? Now, these are not replicated in designs. I haven't run these through animals, but basically, the model was fit against a lot of animal data. And so one would anticipate, or I would anticipate that, this is indeed what would happen. And so I'm going to ignore the first two, particularly the MP allowable, because it's way off. Okay, I mean it's not reliable. I really would encourage you not to look at that. The NEL is closer, right? It's just the way that we represent how much milk can come from that has the problem. The supply is fine. And the MP supply is fine too. It's just the way we're representing that. It's representing it for MP as a linear function, and I just spent about 20 minutes showing you how it's not linear. This nutrient predicted milk is provided in the table, and that's what I would encourage you to use, because it's that integrated equation looking at the amino acid and the energy supplies. And so you can see I didn't really get much additional milk. But when I look at the milk protein that's predicted from the nutrients, I held the same when I lowered the protein in the diet, and then I actually got 40 grams of milk protein by adding in the protected amino acids. And there's a couple of those that are at least methionine that also has a positive effect on milk fat. It's not large, but you can see I lost a little bit of milk fat by probably adding more fat to the diet. And then I gained most of that back, not quite all, by adding those amino acids. So that last 3 slides are sort of my, quick sort of demonstration on how I use the model to try to adjust diets. Now, I normally would not do this by hand in general, okay? Because I'm a firm believer in we got about 30 nutrients we got to worry about, including a bunch of amino acids now, and that's what optimizers are made for, okay? I could spend all day trying to twiddle these around to get them all just like I wanted them, or I could let the optimizer do it in a minute or two, and it's likely going to find a better solution than me anyway in terms of cost and value. So to summarize, not all models that we use in the field are equal. Certainly a lot of the software is based on other models. And so if you're using, those other models, just recognize that they have flaws. And I'm not saying the NASA model doesn't have flaws as well, but it is, one of the most extensively tested model that we have available. The exception might be the INRA model, which is, now a few years old. They took a similar approach as what we did in terms of using a lot of data, a lot of metadata to work everything out. And so, if I were going to trust another model, I might trust the INRA model because it has had so much work done on it as well. But I think, the NASA model, it's the newest one. It's probably the best model currently for NE, MP, net protein, and amino acid predictions. You can use that model, and you could probably use other models as well to successfully feed 15% crude protein diets. There's certainly a lot of examples of that in the field. We fed 15% protein diets to 100-pound cows and not lost production before. The one thing you need to watch for, and when we lose production on these low protein diets, because we do a lot of 14% protein diets to make sure we have a negative control, they always lose dry matter intake, okay? And that is the first thing that happens. And I think that's your thing to watch. If you're messing around with the protein in a pen of cows and lowering it and you see dry matter intake go down, you better reverse right away, okay? Because obviously milk protein and milk production is going to follow that dry matter intake down. So there's something going on, with protein. The common thought is it's an RDP deficiency and we're not getting enough fiber digestibility, but we've never been able to demonstrate that on these low protein diets. When you look at the rat and the mice data that are there's a lot of, there's a sensing center in the brain for amino acids. And when the diets for right mice and rats get messed up in terms of amino acid supply or the ratio of amino acids, they reduce intake. That's how they deal with it. So I think what we're seeing here with these diets, that fall below 15 and we start losing dry matter intake, I think it's an intake center problem. I don't think it's an RDP deficiency problem. There's way too many examples in the literature where, diets were RDP deficient, intake went down, and there was no difference in fiber digestibility. One of the things that you're doing when you're reducing protein in the diet is you're relieving formulation space, and that's really valuable in early and high cow diets because those are very tight formulas. If you can take out some protein and keep the MP supply the same and substitute in starch or substitute in fat and get more energy in the diet, you're going to get probably a lower cost diet because you relieve formulation space and higher production both. Okay, so don't discount the space that crude protein is taking up. Don't put in more RDP than you need to. Okay, I think 9% is enough. Firkins would argue that 10% would be better. It's a linear response. You're going to lose a little bit of microbial protein by going from 10 to 9, but you can make it up with MP, and it relieves formulation space. And the microbes aren't terribly efficient at transferring nitrogen, right? So they lose about 20% of it in converting it from feed nitrogen to microbial nitrogen, and then they're only 20% digestible and only 20%, and they're 80% metabolizable. So we lose valuable or available nitrogen or usable nitrogen by going through microbes. They're good for us. We got to have them. We got to have fiber digestibility, but we don't have to have them maximized. Okay, we just need to have them at adequate levels. And when you're looking at RUP sources, forages are particularly poor. We've got a couple papers where we use that isotope approach and showed that they really just don't. They provide a lot of RDP, but they don't provide very much RUP. Most of your RUPs come from soybean meal and bypass protein sources. So if you want to add RUP, which I think is valuable to do in general in terms of, keeping your RUP levels up so you keep MP up, choose medium and high crude protein ingredients with low cost per kilogram of RUP. Those are going to be the best for fitting into your overall diet. And, I think, use the NASA model. I realize, we don't have software built around it. It's, I think, being put into both AMTS and NDS as a calculator. I'd rather see it being able to be used with their optimizers, and hopefully we can achieve that over a longer period of time. But, use that and use the nutrient allowable predictions. That NEL and MP allowable, they have severe slope bias, and are not terribly reliable. Feed for those amino acid efficiencies around the target efficiencies. And RPA are cost effective when supplementing a bypass protein strategy. We got them to use those several amino acids on a least cost formulation basis, even when we were pricing them at 10 to 15,000 a ton. So they look expensive on a gram basis, but they relieve a lot of formulation space and make it easier to make the diets work and make the diets cheaper. And you don't need to be right on those targets, okay? I mean, within 1% or 2% is probably fine. They can compensate. They have lots of mechanisms that allow them to compensate for deficiencies. So with that, I'll stop, and I'll be happy to answer questions. I'll stop sharing this window, and I can open up the other window, the projected one, so that I can show slides if people want to see them. All right, so looking at the Q&A, no questions on that according to mine.
I don't see any either, Mark, as I'm looking at this. But maybe I could start off with one while others are submitting. Again, if at the top of the screen there's a Q&A chat window, if you all would like to start a discussion there, that would be great. How did you determine the levels of amino acid additions with this research? They're different, right? So with methionine, histidine, lysine, different levels of amino acid in grams per day than leucine, isoleucine, for example. How do you determine what the right level is?
I think there were some limitations on how much we could do with the grain mixes, right? Because they were actually fed through robotic, the automated feeders on robotic milkers. But what I recall is, Don, that we set the MP level at about I think it was an 80% sufficiency based on the titration data I showed you early in the study or in the slide set. And then I think the second level is what we set for hitting the NASM efficiency number. So the first increment would have still been below efficiency, above target efficiency. And the third one would be below target efficiency. The fourth one. The third one would be at target efficiency. In general, we didn't exactly hit those, but that was what our goal was.
Okay. And then relating on Peter Yoder's research in 2020, what do you attribute to the additive response of all 5 infusions? Is it a structural thing, or is it more of a signaling benefit that you're getting out of adding all 5, or do we know?
Yeah, I mean, we have some cell culture papers that demonstrate that clearly. Okay, so what we know about mTOR is that it has a common target on the ribosome that's basically the gas pedal, okay, or the brake, whichever way you want to look at it, or maybe gas and brake together. I don't know, but it will send a signal to the ribosome that's an integrated signal, and it has sort of different sites for those 3 different arms of that I showed you of control. One coming from amino acids, one coming from energy supply in the cell, and one coming from hormonal signals. And those 3 are all sensed on mTOR, and then they are integrated into a common, how much do I press the gas pedal, or how much do I press the brake pedal? And because those are separate signals coming in, being integrated into a common signal, they appear to operate independently and additively. Now, there's some upstream question marks yet that it looks like there may be some crosstalk between the pathways that might reduce some of that additivity. In other words, introduce some crosstalk that would mitigate part of the signal, but that hasn't really been clearly demonstrated yet. We need more work to work that out.
Okay, I did thank you. I did see through the my personal chat window. I had a message come through, so I just posted that. I'm not sure if they all are unable to hit the Q&A chat window here, but I did just send you one. Are you able to see that one?
I've got one. I've got one from Andre Pereira.
Yes.
And he asks, although lower RDP, at 9% could predict more response, would higher RDP at around 10% be able to maximize microbial protein, be more cost efficient? Or is there another variable we should look at instead? That's a really complicated question to answer with a definitive answer, okay? I don't, because we're doing optimization most of the time, I don't pay any attention to RDP, because if you feed more RDP, you will get more microbial protein. I'm sure that the microbial response to RDP will plateau at some point, particularly if you hold energy the same. It definitely will. Okay. But within the data that we have, which ranges from about 13 or 14% RDP down to about 4 or 5, it looked almost perfectly linear all the way through there. The more RDP you add, the more microbial protein you get. So there isn't really a point where you're going to maximize microbial protein. You're just going to get more. But again, to my probably fairly rapid explanation, that when you pass protein through microbes and into the animal as MP, you're only capturing at best about 50%, okay? So if RDP is half the price of RUP, you've probably got an equal weight trade-off, right? Add more RDP or add more RUP, it won't matter, right, cost-wise. If RDP is less than half of RUP, well then yeah, you probably should add some more RDP, recognizing the animals are going to lose on nitrogen efficiency because they're going to be excreting about 50% of that. And if RUP is less expensive than RDP, then you should be adding RUP instead. And you don't probably want to go below 8% RDP. I think 9 is very safe. 8, at some point at 8 or below, we're going to start losing fiber digestibility. And if you do that, that will not work, okay? That will not be a good solution for the cows. So that's my answer. I don't think there's a set number. I mean, RDP tends to be cheap in the marketplace. And so that's why I say don't pay that much attention to it. We don't hold it down. So you will see at times that we will have diets that are higher than 10% on RDP if we're only going for least cost. If we're trying to do a perfect design, then we're going to cap it someplace. And we typically cap it at 10, just because I don't want to have Firkins telling me how much I underfed RDP and that's the reason why my study didn't work, okay? Tongue in cheek. Jeff and I are good friends, okay? We'll argue about this a lot, but I think we both see it mostly the same way, but maybe his answer would be slightly different than mine. So is that the question, Don, or do you have another one?
No, that's good. There was another question Steve Martin posted. You mentioned looking at a protein to energy ratio might be helpful. If we use mcals of Me as a denominator, what would your choice be for the numerator? Total essential amino acids or just MP?
I'd probably use, I mean, we use total essential for pretty much everything in the model. So they're highly correlated. I mean, you can get the same answer largely between the two. But it doesn't solve the variable efficiency problem, right? I mean, it might put it, it narrows the variance a little bit. So you take away some of that. But I think you have to look at it. We would have to look at that. Well, let me back up. We did look at that as an alternative to the milk protein prediction equation that I showed you. Helene looked at that and Roger looked at that a lot, and it does not give better statistics. The actual prediction had lower precision than did the one that we ended up with. Now, is it a lot lower? No, it's still pretty good. I think if I remember right, it was like 15% prediction error versus a little bit under 14% for the one that we used, versus 25 for minimum of NE and MP allowable milk. Okay, so it's still a pretty good equation, but it's not as good as the one that we have, and the one we have does consider energy. So again, if you're using an optimizer, just let it run. Okay. So I was told once, when I was in graduate school that, if you get done and there's no questions, there's two possible reasons for that. One is you did such a lousy job of talking, nobody can even figure out what they want to ask. Or two, you did such a good job of talking that nobody really has any questions left.
I'm sure that's not the case there. I've got another one from Steve.
Okay, Steve, yeah. Steve just posted, yep. Does, the extra detail, sort of address maybe, or what, I think maybe the question is, okay, well, should we still be worrying, given all this extra considerations that we have for amino acids and integrated energy supply, should we still be paying attention to the lysine methionine ratio? And I wouldn't say definitively no, because again, back to my comment about that intake regulation, we know that, we can't have, for example, let's say if, I actually haven't looked. I think the target numbers for metabolizable lysine and metabolizable methionine supplies when you're at the target efficiencies are not quite 3 to 1. I think they're 2.7 to 1 or something like that. But if you're, want to look at a whole range of diets, we can't go, for example, to 1 to 1 and anticipate that the animal will still eat normally. In other words, it won't drop dry matter intake. Nor can we probably go to 6 to 1 and expect it to continue to eat properly, just based on the rodent data that we saw, where that's how they fixed their problem, right? They say, okay, this is really bad protein. It's so bad, I've got to get some other amino acids out of my body stores to try to fix this. So what I'm going to do is I'm going to eat less of this, and I'm going to start degrading body protein, and I'm going to let that backfill my needs for amino acids. And so we have to anticipate that, they're both mammals. There's on all these cell signaling stuff, all that stuff is conserved all the way through from yeast all the way up through cows and everything else that we've checked. So I'm guessing the intake center is the same. So we have to pay attention to, I think, to that ratio, maybe more from the standpoint of intake, but I'm sure also the mammary would, it has limits on how far it can go in trying to mitigate those differences. So 4-to-1's probably okay, 2-to-1's probably okay. Once you get beyond that, then who knows, okay? But don't spend like an hour trying to get your diet down to, 3.2. 3.21, okay, versus you got 3.0 and you're trying to get it to 3.2. I don't think that's probably worth the time and effort.
While we wait for other questions, do you think there's some room yet for improvements of the equations, especially the kind of the the curve relationship between additive responses and relative levels of amino acids? Do you think there's more improvement to understand those? Do you think if they're very well understood and we don't have a lot of room to improve on that front?
I think there's room to improve because we didn't have enough data. There really isn't any studies, or very few, that looked at interactions among amino acids. And so, we don't really appear to have any drastic ones between methionine and lysine, which have had the most work done on them. But that doesn't mean there isn't negative interactions or positive ones in between other ones. And so, we need more work in terms of experimental work to assess potential interactions among amino acids. And it could be among other nutrients too, right? I mean, we sort of like compartmentalize all this stuff and say, okay, well, we only need to worry about protein or amino acids, and we won't worry about energy, or we won't worry about fat, or we won't worry about starch itself, or fiber, or anything else. And we did see that negative impact of fiber, that it just doesn't generate probably as much insulin signaling as starch does. And fat probably wouldn't either, but there probably isn't enough deviation in fat to pull it out for that. So I think there's more room for improvement.
Okay. Well, I don't see any other questions here, unless you see something on your screen.
No.
So yeah, I'd like to thank you, Dr. Hanigan, for spending the time with us today. We really appreciate your dedication to improving, our understanding of dairy cattle nutrition, and really look forward to your continued work in the area. It's very helpful. Also want to let everyone know that attended, they are eligible to receive 1 continuing education credit for participating in the webinar. You can search the ARPAS website for the ARPAS CEU titled Amino Acid Nutrition Webinar. So if you'd like to do that, that's available. And I also want to share with the group that Novita is planning to host additional webinars with a specific focus on better understanding how to improve the use of both the linear and nonlinear optimization systems. We know that they're used, but maybe not as widely used as what we would think they would be, and maybe should be. And so we're trying to better understand what are some of the challenges with it. And so we're assembling some groups to help us better understand that, and maybe even work through some formulations as well through a webinar. So once the plan is finalized, we'll be sending out invitations with dates and times and presenters. So just again, thank you for attending, and thank you, Dr. Hanigan, for presenting today.
My pleasure.